Alright, so we are continuing by talking about the neuromuscular junction. So the neuromuscular junction is a very, very specialized site where a motor neuron, so a neuron that is controlling movement, is going to meet the muscle fiber itself. This is where that muscle fiber first starts responding to that signaling from that motor neuron, and then every single skeletal muscle fiber in that skeletal muscle is controlled by that same motor neuron at the neuromuscular junction. Right? So one muscle is controlled by one nerve. Right? So this is a microscopic image of what that neuromuscular junction can look like. And so we can see here a nerve, and then we've got multiple neuromuscular junctions, but each of these junctions are controlled by that one nerve. So it's going to branch out to each of those junctions, and there's going to be special signals and stuff that are released in those junctions that we'll get to a little bit later. Right, but those fibers are controlled by those neuromuscular junctions, and there is not a different nerve for every single muscle fiber. All right, so we'll talk more about that neuromuscular junction and what actually happens at the neuromuscular junction a little bit later this week, but for now we're going to look at skeletal muscle fibers. So muscle cells are also referred to as muscle fibers, so those two terms are interchangeable because the muscle cells are very long and cylindrical shaped, so they do look like fibers, which is why that name has come to be. And I think was actually the original name before we've realized they were actually cells. So structures of skeletal muscle fibers include the sarcolemma, sarcoplasm, myofibrils, sarcoplasmic reticulum, and T-tubules. So some of these terms might sound slightly familiar from earlier lectures about cells, and there's a reason for that. So the sarcolemma is a special name given to the cell membrane of skeletal muscle fibers. So this is still that plasma membrane that we see generally in cells in the human body, but it's uniquely designed for muscle cells in this particular case, and so it has a special name, which is the sarcolemma. All right, so then we've got sarcoplasmic reticulum listed down here. This is similar to endoplasmic reticulum, except again, it's very unique to muscle cells, and so it gets a special unique name, the sarcoplasmic reticulum. We'll look at this a lot more when we go over how muscle contractions actually happen, but this is very, very, very specialized, smooth endoplasmic reticulum that's going to store, release, and retrieve calcium ions because calcium is really, really important for muscle signaling. We'll talk more about the terminal cisternae a little bit later when we get into more of the actual physiology of how muscle contractions happen, but the terminal cisternae, this is an enlarged region of that sarcoplasmic reticulum that's going to contain really, really high concentrations of those calcium ions. T-tubules is the next thing down here. T stands for transverse, so these are tubes that run in the transverse plane, and so these are going to make sure that the membrane, so this sarcolemma, can get very, very close to the sarcoplasmic reticulum in order to make sure that that calcium exchange is happening efficiently. Right, so you can see that there's these depressions in that sarcolemma that are connected into those T-tubules, and there's a reason for that we'll get into a little bit later. All right. Okay, so the other terms that were touched on, the sarcoplasm, this is cytoplasm specifically for muscle cells rather than the kind of general cells that we talked about a few weeks ago. Myofibrils are going to run the entire length of the muscle fiber attaching to the sarcolemma. which is made out of a bunch of myofilaments. We'll talk about those in a little bit, but this entire unit here that you're seeing is a myofibril. We'll get into myofilaments a little bit later. But this is containing, again, that sarcoplasmic reticulum, the terminal cisternae, and T-tubules. Those together are called the triad. Tri means three, so these three things together are the triad. Really, really important for muscle contractions again. All right, let's talk about the sarcomere. So the sarcomere is what gives skeletal muscle fibers their striated appearance, so that lined appearance because of how myofilaments are arranged. That sarcoplasm is going to have myofilaments. So when we talked about cytoplasm with cells earlier in the semester, there were a lot of structural supports to the cells, so that framework and stuff like that. In muscle cells, these are specifically called myofilaments. Anytime you see that prefix myo, so this right here, that is usually going to refer to something having to do with skeletal muscles. And so the myofilaments that we find in skeletal muscle are actin and myosin. And actin is sometimes referred to as thin filaments because they appear much thinner, and myosin is sometimes referred to as thick filaments because they tend to be much larger in diameter, the way that we view them. So these myofilaments, together with some regulatory proteins called tropomyosin and troponin, make up the sarcomere. All right. And the sarcomere is basically the functional unit of a muscle fiber. So this is a massive chain of thousands of repeating sarcomeres that are going to make those functional units of the muscle fibers that are forming the myofibrils. So this is what a sarcomere looks like with our graphical representation. And so here we've got that entire muscle fiber. Right here we've got those multiple nuclei scattered throughout, and then we can see here in this diagram that lined appearance, so that striated appearance that I've mentioned before and we just didn't get to see very well in the microscope image. Lots and lots of mitochondria are going to be present in muscle cells because we need lots and lots of energy available. We've got that sarcolemma surrounding all of those myofibrils, and this down here at the bottom is what zoomed in view of that myofibril is going to look like. And so you can see right here that sarcomere is that basic contractile unit, and then we have lots of these linked together end to end. And then these are all going to be covered by that sarcoplasmic reticulum and then the T-tubules and stuff like that. So we've got the thin actin filaments here represented in green, and then we've got the thicker myosin filaments that are colored more of a purplish here in this image. And so where those actin filaments are coming together and linking up together, you can see that they're making this kind of zigzag structure that we call the Z-disc. And then we've got where some of those myosin filaments that are starting to overlap with other structures, that's called the H zone. All right. And then we've got the I band and A band. So the I band is the distance between the ends of those myosin filaments, and then the A band is the actual length of those myosin filaments themselves. And then that little section of those other striations here in the middle that are going opposite directions from the actin and myosin, those are called the M line. And each of these have their own special functions that we'll get into a bit more detail later. But that M line is going to basically move as these muscles contract. All right. So this is how we get that striated or lined appearance here. So those A bands, so this section right here with those thicker myosin filaments, is going to look darker than the lighter I bands from the actin filaments that are much, much thinner. And so that's how we get that lined appearance of muscle fibers for skeletal and cardiac muscle tissue. This does not exist in smooth muscle tissue. So myofilaments here, zooming in a bit more to that sarcomere and kind of removing some of that excess tissue out of the way so that we can see just the actin and myosin filaments. So H zone here, we've got the M line right there, Z lines, and then we've got the I and A bands made up of the actin and myosin filaments. And so those myosin filaments, they have all of these little structures on them called the myosin heads. And this is where actin is actually going to bind to that myosin during contraction. These are blocked off during relaxation of muscles. And then over here we've got those actin filaments, much different structure. They do not have those heads on them, but there is going to be binding sites for myosin. So those actin binding sites here on the myosin heads are going to bind down in there. And we've got all these repeating actin subunits. And so on actin, you can see that troponin and tropomyosin that I mentioned earlier. So these are proteins that are regulating muscle contraction. So you can see that the tropomyosin is this kind of orange band here that's actually blocking the binding sites for myosin. And that's really important during relaxation of muscles, because if we don't block that binding, then they're going to stay bound together, which means that the muscle is staying contracted, and that can be a really big issue. All right, and then troponin is going to just kind of help with regulating that entire process as a whole. We're going to continue talking about excitation-contraction coupling in our next video.